Okay, this lecture is Brain Neurons Part 1. We're going to be talking about normal synapse function, where two neurons communicate with each other, and some of the basic changes that can happen with that in special situations. So starting out with the anatomy of a neuron, this is a normal neuron. Its nucleus is up in this location. Where the nucleus is located is called the cell body. Then it has a long tubular segment called the axon. At the beginning of the axon, that's called the axon hillock, and that's where an action potential starts, where the neuron fires an action potential. The fat wrapped around the neuron here is like the insulation around an electrical wire, and it speeds up conduction because, in a sense, the conduction can jump from the spots in between the fat, the myelin. As you put more myelin on a neuron, its conduction becomes faster. So like when you learn a sport, how to hit a tennis ball, you keep hitting against, let's say, the ball machine, and by the second week you got more myelin on those neurons and you can hit the ball faster. And they're made by cells called oligodendrocytes. These myelin is what is damaged with multiple sclerosis, slowing down neuronal conduction, and it'll eventually get to the point where it injures the nerve in multiple sclerosis. The action potential is going to travel down this axon towards the axon terminal. The axon terminal is of the presynaptic junction. This is the synapse. Synapse is the space between the pre and the postsynaptic neurons. So this is the one that's sending a message by its neurotransmitter vesicles are going to fuse with its synaptic membrane and then release neurotransmitter to travel across the cleft and to bind with the postsynaptic neuron and then have an effect on the postsynaptic neuron. So that's standard normal anatomy of a neuron. Over here is where this neuron receives messages through its dendrites. So that's sort of, in general, the receiving end of information. And then the neuron sends information through its axon, and then it can communicate with the postsynaptic neuron in that way. Okay, here's a standard anatomy of a serotonin synapse. So serotonin neurotransmitter, there's a theory that actually serotonin is decreased in persons who are depressed. That's actually never been proven. It's more of a marketing thing than anything. But just for you to see an example of a serotonin synapse, because it's a very commonly talked about synapse. So the serotonin, it's made from tryptophan. The serotonin then is in the synaptic vesicles. When an action potential is coming down its axon, calcium enters a neuron, then the serotonin vesicle fuses with the plasma membrane here, and it releases serotonin across the synaptic cleft. It travels across the synaptic cleft, binds with the postsynaptic receptor, and then thus exerts an effect on the postsynaptic neuron. Afterwards, the serotonin then will start to diffuse back towards the initial sending neuron, presynaptic neuron, and it'll have a serotonin reuptake transporter. Sometimes it's called a SERT, S-E-R-T. And then it can go into the cell. It can be put back into synaptic vesicles, or it can be degraded by a monoamine oxidase enzyme. And then the other thing you're going to hear a lot about is selective serotonin reuptake inhibitor, SSRIs. That's a very common type of medication that is called an antidepressant and is used to block this reuptake transporter. And you can also have something called SNRIs, serotonin and norepinephrine reuptake inhibitors that'll block both of them rather than just one of them. So this is a basic synapse, and just this idea of reuptake being inhibited, leading to more transmitter in the synaptic cleft, that's a very important concept for neurons and neuron-related medications. Okay, this next synapse is an example of a dopamine synapse. First of all, we'll show a normal dopamine synapse. And so initially, I got flies buzzing around me. You'll have an action potential coming down the axon. And I drew the charges in this way because normally you have a negative charge, about negative 65 millivolts inside the neuron. But when it's depolarized, you'll get a positive charge transiently within the neuron, and you get the negative right adjacent to it. Calcium then, when the action potential comes down to the synaptic terminal, calcium then, these voltage-gated, VGCC just means voltage-gated calcium channel will open up because of the depolarization. That's why it's called voltage-gated, because it's the charge of the neuron when it becomes positive. And then the calcium will enter the neuron, and it'll cause the release of these dopamine vesicles. So the dopamine is a neurotransmitter. These vesicles will fuse with the plasma membrane of the presynaptic neuron. They'll release dopamine across the synaptic cleft. Dopamine will bind with this DAR, that stands for dopamine receptor. These are actually more precisely type 2 dopamine receptors, which if you ever become interested in studying dopamine in more detail, that's a very important type of receptor for dopamine, type 2. It binds to a plasma membrane, what is called a G protein. So these are what is called G protein coupled receptors. Neurotransmitter receptors, the most common things we think of are they're either coupled through a G protein, for example, or they're just open up an ion channel. So with this dopamine, it's a G protein coupled receptor. And that's normal dopamine transmission. And now we're going to contrast with abnormal dopamine synapse, and this is after an AP is given. AP stands for antipsychotic medication. Antipsychotic medications are also called neuroleptic medications. So what's happening here is this antipsychotic would be an inhibitor, and it's inhibiting dopamine transmission because it's binding to the dopamine receptor and blocking it. So I drew an X through the dopamine receptor that it's blocked. So here's AP for antipsychotic, blocking it here. Here's another AP for an antipsychotic, blocking it here. And then the reason for drawing this neurotransmitter, this synapse, is to show what's happening. The postsynaptic neuron will try to compensate for having diminished dopamine, and it'll produce additional dopamine receptors. Notice over here we only had 2 of them. And by this same scale, we've now made many more. So it's trying to increase its sensitivity to dopamine, the postsynaptic neuron. Presynaptic neurons can do different things to adapt to a drug trying to change their normal metabolism. They might, for example, make more synaptic vesicles, which I have drawn here, more dopamine synaptic vesicles. And so the point I'm trying to get across here is that the brain is not stagnant. When a medication comes in that's changing how the synapse works, the brain, in a sense, fights it. It tries to make more receptors on the postsynaptic neuron. It'll try to make more synaptic vesicles on the presynaptic neuron. It's going to try to do something to reestablish itself back to normal. That's called homeostasis. The brain wants to function like it normally does. It does not like a drug coming in there. Okay, here's just a quick trick to memorize, the common neurotransmitters. Like the word damn is often euphemistically said dang. So just say dangs, D-A-N-G-S, and there's a couple of the A's, and you can real quickly just memorize all the names. The reason why you'd want to do this is if you wanted to memorize those. For our purposes now, we don't need to, but I'm just showing you a real quick trick. This is like typical what I would do in school. I'd make a mnemonic to memorize all the names, and then I would start adding all the information to the mnemonic, walk around and talk to myself. I call that a walk and talk method of study, and it's real helpful for memorizing stuff. Okay, let's see, we got a little bit more here. Oh, these are some things. We briefly talked about these before, but it's just a quick refresher because we're going to subsequently, in a different lecture, get into more advanced synaptic concepts. So this is the potassium sodium ATPase. I like to put the potassium first, and the K stands for kalium. That's Latin for potassium, for the potassium coming into the cell. So 2 potassiums are pumped into the cell. This is active transport because it depends on an ATP. So a phosphate is pulled off of the ATP. So we're using up this ATP to run this potassium sodium ATPase. The N is for natrium. That's the Latin for sodium. Notice that 2 potassiums are pumped in, 2 positive charges, and 3 are pumped out. What that means is we're pumping out positive charges, the net effect, and this contributes to the resting membrane potential of the neuron being negative, about negative 65 millivolts. There's actually a little bit more to what causes the resting membrane potential, and there is such a thing as a leaky potassium channel that contributes to it. But for our purposes, we need to know that the potassium sodium ATPase supports this resting membrane potential, and it generates a gradient whereby there's going to be a lot more sodium outside the cell as it's being pumped out in these large amounts. And also because there's a negative charge inside the cell, that this sodium that's sitting around in the extracellular matrix is going to really want to come into that cell. And the energy of running with that electrical chemical gradient of sodium is going to power other things. For example, to pump calcium out of the cell, which is very important to do, there's something called a NACA exchanger. Na for Na for sodium, Ca for Ca for calcium exchanger. So it's often abbreviated NCX, sodium calcium exchanger. And that can pump many thousands of calciums out of a cell very, very quickly. So after an action potential and you need to get the calcium out of the cell, the knockout exchanger will crank it out real fast. And things that will disrupt this process, first of all, if a person eats excessive dietary amounts of sodium, the ions are going to be balanced out and it's going to lead to increased sodium inside the cell, and that's going to weaken the electrochemical gradient. It's going to weaken the ability of the cell to do work. So it's bad to eat excessively high amounts of sodium. Second thing is you need some magnesium in your diet because magnesium is bound to the 2-phosphates on the ATP. TP means triphosphate, and it helps to stabilize those negative charges. So if you're deficient in magnesium, your brain cells are not going to run as efficiently. And if you're deficient in potassium, your brain cells are not going to run efficiently because it'll tend to be replaced. Its net amount in the ionic balance of the neurons is going to be replaced largely by sodium. So you don't want that. And the point of that too is potassium and magnesium, they come from plant foods. If you eat plenty of plant foods, you'll have all the potassium and sodium you could want, and your neurons will run better. Plus, you'll avoid the processed food that tends to load you up on the sodium, which leads to problems with your neurons. Okay, so I'm making the point that the plant diet gives you what you want. It gives you the potassium and the magnesium, and it minimizes sodium. That's what we run best on. Okay, now here's a little bit more complicated neuron, and we don't need to go into all the detail of this, but I just want to mention this is a glutamate neurotransmitter type neuron. Glutamate is the most common neurotransmitter in the brain. There's different articles will say different things, but in general, it's about 75% of brain neurotransmitters. It's an excitatory transmitter, meaning that it excites the postsynaptic neuron. We talked about the KN, potassium sodium ATPase pump. We talked about pumping calcium out. We talked about the negative 65 millivolt resting membrane potential of this neuron. So this is a glutamate neuron. This is typical in the hippocampus, the memory center of your brain, perhaps the most important part of your entire brain. And the relevance is there's a lot of things that can increase glutamate transmission. Glutamate transmission is increased by stress. It's increased by potentially by MSG, potentially by MFG, stress equivalents like caffeine, sleep deprivation, corticosteroid medications, and there's other things that can potentially increase it. So that's why I'm a big believer in don't be drinking any caffeine, because you've already got things putting you at risk to ramp up this glutamate. You don't want to do that. You'll have more resilience, you'll be healthier, your brain will function better if you avoid unnecessary elevation of this glutamate transmitter. It's an important part of your memory system. And so we'll come back to this later, but calcium balance is also important in both the pre and the postsynaptic neuron. And the SERCA is the sarcoplasm endoplasmic reticulum calcium ATPase, and that's a pump to pump calcium out of the cytoplasm, because you've got to maintain a low amount of calcium in the cytoplasm, because whenever calcium is in the cytoplasm is elevated, it's like a light switch, and it causes that neuron to fire an action potential. And if you overdo it, fire too many action potentials, the neuron runs out of energy and it can die going into apoptosis. So we'll talk about all these things in more detail, explain them more, but the purpose of this lecture was just to give you a sense of how neurons work, be familiar with some of the terminology, so when we step into the more advanced, more interesting stuff, this will be familiar.